Hello, I'm Dr. Aizaz from medicovisual.com, and in today's visual lecture we will talk about histogenesis of cerebellum, and along with that I will briefly discuss the physiological aspects, the basic physiology of the cerebellum as well. So as we have discussed in the previous lectures that the cerebellum or any of the part of neural tube, it consists of three layers. The innermost, which is near to the lumen, this is, let's suppose here is the lumen of the neural tube. For example, this is the neural tube, for example. So this is the inner side, this is the inner or luminal side, and this is the outer side. So this is the inner or luminal side. So here this is the ventricular zone, which consists of these epithelial cells, and these are the ependymal cells. So with that reference, this is also called ependymal zone as well. And then outer to it is the intermediate zone or mental zone. Then outside is the white matter layer, which is the marginal zone. Another layer of cells that also arise by the proliferation of cells in the epithelial cells in the ventricular zone. Basically, we can say that there is another wave of proliferation of cells in the ventricular zone, which leads to formation of another cell layer like this. So now we have the cell layer. Now what will happen that some of the cells in this region, they will differentiate into a specialized type of cell with this tree-like dendritic arrangement. These cells are called Purkinje cells on the name of scientist who first discovered them. Now these Purkinje cells have this beautiful artistic pattern of dendrites on them, and this pattern of dendrites, this beautiful pattern, is called arbor vitae, which literally means tree of life. Vitae is life and arbor is this tree-like pattern, arbor vitae. So these cells are Purkinje cells, which are differentiated from these cells which came from another wave of proliferation from the ventricular zone. Now these cells, they will develop axons, and before that what happens that cells in this deeper layer of mental zone, this is the mental zone, cells in this region, some of these cells, they will differentiate into deep cerebellar nuclei. In the cerebellum there are some deep cerebellar nuclei deeply placed in the deeper part of the cerebellum, embedded within the white matter. If you don't know, don't worry, you will learn them in neuroanatomy. You will also learn their names. They have alien-like name: emboliform, globus, vestigial, dentate. Don't worry about that. You will learn them. Ultimately, it's not, by the way, important to learn their names in embryology, but you must know that they develop from this mental zone, and here some of these cells, they will differentiate into cells of deep cerebellar nuclei. And as this green one, of course they are not actually green in color, it is just for the sake of representation in this lecture. So these deep cerebellar nuclei, they will release certain chemicals, certain factors which will attract the axons of these Purkinje cells. So these axons of these Purkinje cells, they will come and they will not just try, they will actually synapse with these deep cerebellar nuclear cells. And then some of the cells in this region, some of other undifferentiated cells in this region, they will also differentiate into another type of cell. These cells are called Golgi cells. Don't worry, we will see what they are doing here in the later part of this lecture. By the way, this blue one is the pia mater. Don't get distracted because of this pia mater. So now anyhow, at this time we have three types of cells in this region. One is this Purkinje cells, second is these undifferentiated cells which are still undifferentiated, and third is these Golgi cells. Now what will happen that all these three types of cell, this layer as a whole, it will migrate outward, and of course pia mater and other things, they will also lift it up and they will migrate upward But these Golgi cells, even though they will migrate, but they will not migrate as much as the other cells. So what I am saying that all these will migrate, but Golgi cells will not migrate drastically. One thing. Second thing is that, as we have seen, that the Purkinje cells, they have established the synaptic connections with these deep cerebellar nuclear cells. They will continue this connection. This connection will not be broken actually. This connection will remain there, and their axon, they will sort of lengthen up as they continue to reel out more and more axon. Its axon will sort of lengthen up as they migrate. So as they say, seeing is believing. So let's watch the animation to understand what is happening here. So here is the situation. So as I have told you, the axon will reel out for the Purkinje cell. Golgi cell will not migrate drastically, and the undifferentiated cells, they will also migrate outward. Let's watch the animation again. So here is the situation right now. Now what happens that here some of these cells, although in this model we have ample cells for the explanation, but in reality the cells are not enough. So these cells, they will proliferate further, and you can say cells in this region, they undergo another wave of proliferation. I don't know why books like to write in this type of language, but that's how it's written in the textbooks, and I am reiterating that because some people complain that you do not use bookish type of language. So here I am with sort of bookish language, so that might help you in your exam. If you are writing in theory, you can write such type of language for better marks perhaps. Some professors love to see such type of language written by students. So anyhow, that's what it is. So what happens that cells in this region, they undergo another wave of proliferation, and then what will happen that after that some of these cells in this region, they will differentiate into yet another type of cells, and this yet another type of cells, they are called granule cells, right? So these granule cells, they are basically like bats. And you know why I am saying this, that what will happen that they will form these axons, and then these axons, they will sort of bifurcate like this. So T-like axonal process will be formed. T-like axon, that there is this main stem, and this main stem bifurcates into two like this. So T-like arrangement, this is like alphabet letter T. So T-like axonal process will form, and they are in this region, but their work is in more deeper part of the cerebellum. So what will happen that they will migrate downward, and they will move like this. They will migrate downward, right? So these are like bats, because when bats, they are sleeping, especially, they hang upside down. So this reminds me of bats somehow. I don't know why I have such type of imagination, but anyhow, that's what I think of them. So this is the main part of histogenesis. Besides that, there are some other cells, of course. Some of these cells in this region, they will also differentiate into basket cell and stellate cell. Now what is basket cell? These are the basket cells. So basically their axonal process, they will form basket like arrangement around the soma. Soma is cell body. This is the cell body of Purkinje cells. So these basket cell, their axonal process, they will form basket like arrangement around the cell body of Purkinje cell. If you just focus on this basket, you can see that this type of basket is formed. You see, it's a beautiful process, right? I love teaching it, and I love visualizing it. So these are the basket cells. Then we also have these stellate cells. Here, these are star-shaped cells. The stellate cells, they are slightly in superficial part, and basket cell, they are slightly in the deeper region. They are both of these cells, they are inhibitory, and of course Golgi cells, they are also inhibitory. All these three types of cells, they are inhibitory. But basket cells, they inhibit at the level of soma, cell body, and these cells, stellate cells, they inhibit at the level of dendrites. So basket cells are more, they are more powerful in inhibition. They are more powerful inhibitory cells, you can say, right? Then, of course, here some fibers will arise. These are the climbing fibers. They will come here, and they will they will form lots of connections with dendrites, right? Dendrites of Purkinje cells. And here then we have the mossy fibers. These are like the mossy plant, the moss of the plant moss, and they will form a lot of connections with these granule cells. And these granule cells, they are bifurcating here. And by the way, these bifurcating fibers in this region, they are called parallel fibers. They are running parallel like this, so these are called parallel fibers. So this is basic arrangement. This is the basic histogenesis of cerebellum. You can watch whole of this animation again. That first we have a wave of proliferation forming these cells. Then we have formation of Purkinje cells. Along with that, we have the formation of these cerebellar nuclei and formation of these synaptic processes. Then we have formation of Golgi cells and migration. Then we have formation of granule cells, formation of T-like bifurcation and parallel fibers. Then they will come down, migrate down like this. Then we have, of course, the formation of basket cell and stellate cells. Then we have climbing fibers here, and mossy fibers will be there like this. And one thing I forgot to mention, what is the role of these Golgi cells? Golgi cells, they will form this axonal process like this, and they will inhibit these cells. This, not cells, I mean they will inhibit this connection. The mossy fiber and granule cells connection, they will inhibit this connection. So these are also inhibitory. So this was almost everything about the histogenesis of cerebellum that you need to know. Of course, there are some other cells as well. For example, the Candabellum cells, the Lugaro cells. Then we have special hair cells in this region. So there are lots of other cells as well in the cerebellum, but their origin, their embryonic origin is not clearly known. Number one. Number two, their function is not precisely known. So because their embryonic origin is not clearly known, and as they say, these are not high yield topics, so I will not go into their details. This much information should be enough for most of you people watching this video. But now I will discuss a few uncertainties, ambiguities, and some caveats as well in this lecture. And let me tell the I think ambiguities of my own lecture. So I am being the critic of my own work right now. So if I go to the first part of the first model, you see I have created these mental cells of the mental zone, these neuroblasts, and this marginal zone. Now, the thing is that typically the neuroblasts, they are somewhat differentiated, right? Neuroblasts are somewhat differentiated, and neuroblasts have these dendrites, and they have this axonal process which are going to form this part, what we call marginal zone, white matter of marginal zone. So they are somewhat differentiated. Now, this much degree of differentiation, it's a bit difficult for them to form such type of specialized cells like deep cerebellar nuclei and things like that. So it's a bit ambiguous. That whether they are the typical neuroblast in this region as in other parts of neural tube, or they are specialized sort of less differentiated type of neuroblast in this region. This is not known in literature. I cannot find any reference, so that is why I haven't shown such type of axonal process. But then what is this? What the hell is this that I have created? I have created these fibers. I have created these fibers because they are still mentioned in the textbooks. Personally, I am not even satisfied from my own lecture. I think this is not really good work of mine. This is not my best work. I am a bit sad about it because unfortunately I cannot find significant information about this topic. I cannot find the crystal clear concept of this topic from the textbooks, from the research article. I tried digging the 1960s book, I tried digging the 1930s book, of course, besides the latest modern books. I tried digging very old books and I tried studying old and new research articles, but few things I am still not clear about. So I am going to mention those things in this part of the lecture. If it's too boring for you, you can skip this part, but I am still gonna mention these, and I am gonna mention the ambiguities and uncertainties, or you can say pitfalls of my own lecture in this part, right? So one thing I mentioned, that I am not sure that these are typical neuroblast or these are some special less differentiated type of neuroblast. Second thing that many textbooks they are doing is that they say, here you see there is another wave of proliferation and there is formation of these cells here. Now some textbook at this point they say that, okay, let's call this layer internal germinal layer. So okay, fair enough, we call it internal germinal layer. Then they say, okay, it's internal germinal layer. So this internal germinal layer, it is going to form the Purkinje cells. It is going to form the Purkinje cells. It is going to form the Golgi cells, right? Golgi cells. So then what happens, that now the thing is that now these cells they will migrate outward. And now this internal germinal layer, it is no more internal, now it is external germinal. So internal germinal form the Purkinje cell and Golgi cells. Now this is the external germinal layer, external germinal. Now they call it external germinal because these cells, they underwent another wave of proliferation to form more cells in this region, which is basically the external germinal layer. Now this external germinal layer, now this is going to form the more cells, I mean the granular cells, the basket cells and the stellate cells. The basket cells and stellate cells, of course. So this is very clear. Some textbooks, for example BRS specifically, they mention like this, that internal germinal layer is there, it is going to form the Purkinje cells and Golgi cells. External germinal layer, it is going to form the granular cells, the basket cell and the stellate cells. That is very clear. But then there is another twist. Some textbooks, they come up with something very strange, that they say that external germinal layer is going to form Purkinje cells, external germinal layer is going to form basket cell, and external germinal layer is going to form stellate cells, and the internal germinal cell, it is going to form the granular cells, granule cells, right? And even Golgi cells. So what the heck is this, all this confusion? Even I don't know, to be honest. This is sort of a discrepancy among the literature, among the textbooks. But what I think of it is that actually as these cells, as they moved here, these Purkinje cells, as they moved here, maybe Now they are thinking that okay, now they are external germinal layer. So ultimately external germinal layer, as they migrated here, ultimately they have formed the Purkinje cells. I think there is no point of demarcating the histogenic process into the external germinal and internal germinal layer. This is very confusing and this is absurd classification. I don't agree with this. You can simply say that of course they develop from the LR plate, and basically they develop from the cells of LR plate. You can say that, and that should clear the confusion. The good thing is that fortunately this topic is not very high yield. It's not very high yield topic, the histogenesis of cerebellum. I'm specifically talking about that. The previous topic, the gross development of cerebellum, that is somewhat important, but the histogenesis of the cerebellum, it is not very high yield topic. So hopefully you won't get much questions from this topic. But anyhow, this was some confusing points that I wanted to tell you. And yeah, finally, before concluding this part of lecture, let me tell you that histologically I should say, that the cerebellum, it is actually divided into several different layers. We have these white matter, which consists mainly of parallel fibers. So this parallel fibers, along with that the arbor vitae, and what is this, basket cell, stellate cell, arbor vitae of Purkinje cells, all this, they constitute a single layer which is termed as molecular layer. So this is molecular layer. Then we have a single cell layer thick layer. This is just consisting of cell body of Purkinje cells, and this is called, of course, Purkinje cell layer. It's very logical to call it Purkinje. I think it's a single E here. So this is Purkinje cell layer. And then last one, which consists mainly of Golgi cells, as well as we have the granule cells. Mainly it's a granule cells, and of course we have the Golgi cells. This is called granular layer. And of course finally there is a lots of white matter here, and very deep in the cerebellum we have the deep cerebellar nuclei. Technically it's not a layer, we have the deep cerebellar nuclei in this region. So this is the just a separate, you can say, part of cerebellum. So molecular, Purkinje and granular cells, you can remember it with the help of a dirty mnemonic that I've created, that morning pussies. I'm sorry about the explicit language, but it will help you remember. So again, it's a necessary evil. Morning pussies gratifies. And if you want to remember cerebellar nuclei, you can remember nicely. So morning is for molecular, pussies is for Purkinje, gratified is for granular, and nicely is for nuclei. I mean the deep cerebellar nuclei. These three are basically layered. This is just that you know that it is the part. There is that part of cerebellum that consists of deep cerebellar nuclei, and by the way, it is from outside to inside, from superficial to deep. This is the superficial part, this is the deep part, right? And here there will be the fourth ventricle, lumen of fourth ventricle, right? So I hope you got the concept of histogenesis of cerebellum. And finally, of course, before concluding the histogenesis, let me show you a section of the cerebellum, histological section of cerebellum, like this. So here is the histological section of cerebellum. This is a more realistic three-dimensional model. So outside here you can see lots of parallel fibers formed by the hanging granule cells, upside down hanging granule cells. You can see it clearly, and this is, by the way, molecular layer, which consists of the parallel fibers, the arbor vitae, the basket cells and stellate cells. They are hidden inside here. These are the stellate cells in this region. This is the molecular layer. Then we have this Purkinje cell layer. You can see it in the 3D as well. These are the Purkinje cells, and we have these Golgi cells and granular cells in this region. Of course, we have the mossy fibers and the climbing fibers, right? So we have mossy fibers and climbing fibers. By the way, these are the folias and fissures. The cerebellum, it is not a simple straightforward smooth structure, right? It is thrown into folds because of its large surface area. Cerebellum is not smooth like this, it is like this. There, this is the situation. These upward part, these are called folia, and these downgoing part, these are called fissures. So we have fissures and folia. So here you can see this is the folia, this is folia, and this is a fissure. This is the fissure like this. So three-dimensionally you can observe this in this histological model. I hope you are very clear about histogenesis as well as the histology of cerebellum. You must understood the histology of cerebellum as well with the help of this lecture. I hope so. So are you really clear about this? You can keep watching it to get them imprinted in your mind so that you never forget, hopefully. Right, so now in the next part of this lecture, I will discuss the brief physiology of cerebellum. Now, before discussing the physiology, a bit of disclaimer here that I will not go into physiology of cerebellum in details because the two reasons. Number one, we are not aware properly exact physiology of cerebellum. It is not clearly known. It's more of a hypothetical and speculative mechanisms that are discussed in the books. So the one reason is this, and second reason is that it's a huge topic, and there should be a complete lecture on the physiology that we will create as well when we will discuss the neurophysiology. But in this lecture of embryology, I will discuss the basic aspects of circuitry. So for the sake of explaining the rationale of presence of these cells in this circuitry, I will explain some of the basic functions of these cells. So that is why I will discuss basic level physiology of cerebellum. Again, I will not go into details. So let's suppose this is the cerebral cortex here, and simple Dr. Najib style diagram here, and of course here is the cerebellum. Now here is the motor cortex. Now let's suppose that you want to move your hand and you want to pick a cup of coffee, for example. Okay, let me draw a diagram for you that if you have, for example, a cup of coffee here. This is a cup of coffee that you want to pick. This is a hot cup of coffee, and you have your hand somewhat here. And you want to move your hand here towards this cup of coffee, and you want to pick it. Now, the movement will start with the help of course, there is the role of basal ganglia as well, which contains the motor plans actually that we will not go into detail. Now, the function of cerebellum starts as the movement start. Now, first thing that it should coordinate a smooth movement. Cerebellum should make sure that your hands exactly goes to this cup of coffee, and it should not overshoot. Like there should not be excess movement. There should not be under movement, and there should not be excess movement. There should be precise movement, and this precise movement should be quite coordinated and smooth. There should not be jerky movement, it should be coordinated movement. So your hand should exactly go here, and it should not overshoot. For example, it should come here in this region, but it should not go beyond this. It should not overshoot this point. So these things that the cerebellum should ensure. Now, to be honest, we do not know exactly how it happens, but there are certain hypotheses, or these are more of speculations, and these are not my hypotheses or my speculations. These are speculations of some of the authoritative figures, some of the scientists who propose these hypotheses. So let's start the discussion that we have the mossy fibers, and mossy fibers, they are coming from lots of different sources. One of the important source is cerebellar fibers or corticopontocerebellar fibers. You know, in the pons we have the what is this pontine nuclei, right? So from the cerebral cortex some fibers are coming in this region. And these fibers, they are synapsing here with the pontine nuclei in the pons, and then they will go back into cerebellum. Now these are one of the important component of mossy fibers. Mossy fibers are coming from a lots of different sources, so I will not go into all other sources. One source that is important for discussing the physiology right now is these corticopontocerebellar fibers. These are one of the component of mossy fibers. So these red one, okay, let's draw it with a green color. These are the green one, right? So these green mossy fibers are coming into the cerebellum, let's suppose, and here they will form moss like arrangement, and they will synapse with those upside down hanging fibers. You know what are those? These are the granule cells, upside down cells, granule cells here in this region. They will synapse with these granule cells like this. So they will synapse with these granule cells, right? And then of course they are forming these parallel fibers. Of course there will be other granule cells as well that are going to form the parallel fibers. Now these parallel fibers, they will synapse with lots of Purkinje fibers. You know, Purkinje cells, not fibers, I'm sorry. So they will synapse with lots of Purkinje cells. You know, lots of Purkinje cells are there in this region. They will synapse with those Purkinje cells, those flask-like Purkinje cells in this region. And okay, let's draw a single Purkinje cell here. So now these Purkinje cells, they will send their axon downward into very deep layers, and here they will synapse with what? Deep cerebellar nuclei. So here they will synapse with deep cerebellar nuclei. Now deep cerebellar nuclei, they will send axons back, and these axons, they are coming back with the help of there will be of course relay station in the thalamus here. There is a relay station in the thalamus in this region, and ultimately these fibers form the deep cerebellar nuclei. They will go back to cerebral cortex. And another important thing is that as the mossy fibers, as they were going upward to synapse with the granule cells, they also established a connection, a collateral connection, a stimulatory collateral connection with the deep cerebellar nuclei, like this, deep cerebellar nuclear cells like this. So now what happened? You see, as the movement started, as the cerebral cortex said, okay, let's move the hand. As the movement started, immediately a copy of this motor order was sent via these cerebropontocerebellar fibers or mossy fibers. Copy was sent to cerebellum, and this copy, it stimulated immediately. It first stimulated the deep cerebellar nuclei, and as it stimulated the deep cerebellar nuclei, this deep cerebellar nuclei, it sent a message back to the cerebral cortex. Okay, good going, continue with this movement. So you see, it sent an initial go-ahead signal. You can say it sent an initial go-ahead signal, which sort of potentiated the movement. So initially, it sort of, as soon as the movement started, the deep cerebellar nuclei, it sort of potentiated the movement. It said, okay, go ahead with the movement, and movement continued. But within a fraction of second, within a few milliseconds, what actually happened that these mossy fibers, as they are also sending the signal to the granule cells, and from granule cells Purkinje fiber, the signal was sent to Purkinje cells here, and Purkinje cells sent another signal back to deep cerebellar nuclei. Now the Purkinje cells are inhibitory for the deep cerebellar nuclei. These are inhibitory for the deep cerebellar nuclei. They sort of applied brakes to the deep cerebellar nuclei that okay, stop. A lot of movement has already went on, so now you should stop. And as it stopped, what happened there? There is prevention of overshooting. There is prevention of over movement, right? So as there is the brakes are applied, this signal, this go ahead signal, it sort of inhibited, it got inhibited, and the movement also gets inhibited. So initially there was a movement, but within a few milliseconds this movement has to stop to prevent the overshooting, that exactly at the time of the at the spot of the cup this movement should stop. Now, how does the cerebellum know that what should be the timing? Cerebellum is calibrating a lots of information. It is getting information from the spinal cord, from the different regions of the body. It is from even the eyes, from different regions of the body. It is getting the exact information that where your hand is right now, where is that the cup, and it can calculate on the go. I mean, during the movement, it can calculate that what will happen is if the movement continued like this. It knows the degree of movement that is going on. It knows the strength or force of movement, and it knows that what is the intensity of movement and stuff like that, and it can calculate. But how it exactly does, that we unfortunately do not know. Basic idea of this circuitry, we know that it prevents the overshooting, and the basic mechanism that I've told you, this is the basic mechanism. The exact timing control, how it happens, that I do not know. We do not know in general. I mean, the scientific community do not know in general. So this was the basic idea. Now this was about the mossy fibers. What about the climbing fibers? Actually, the climbing fibers, they are important for motor learning and for the error signal. So let's draw the diagram again, and as you know that in the medulla we have the olivary nucleus. Basically it's the inferior olivary nucleus here in this region. So here is the inferior olivary nucleus, two inferior olivary nuclei in this region. There is a superior olivary nucleus as well that is related with hearing, that we will not discuss, but this is the inferior olivary nucleus. Now what will happen that the cells in the inferior olivary nucleus, they will send axons, and these olivocerebellar axons from the olive, the axons are going into the cerebellum. These are the olivocerebellar fibers, and these olivocerebellar fibers, these are going to form the climbing fibers. And by the way, olivocerebellar fibers are the only source of climbing fibers. Mossy fibers, there are lots of different sources of mossy fibers, but olivocerebellar is the only source of climbing fibers. So climbing fibers or olivocerebellar fibers, they are going to cerebellum. And here let me explain a few things. Now inferior olivary nucleus, as the movement started, the motor cortex, it gave a copy here to the olive, and then at the same time, in the spinal cord has the proprioceptive information. It has the information from the muscles and joint, that what is the position of joint, and real time it knows. The inferior olivary nucleus, it has the real time information of the position of your muscles and limbs and other parts of the body. It has the real time information of degree of movement, the tone and stuff like that. It has the exact information. So now you see it has the motor order and it has the information of exact degree of movement that is going on. So it compares this information. It compares the motor order. It compares the motor order from the cerebral cortex with the actual movement. You can say it compares the intended movement with the actual movement. It compares the intended movement with the actual movement, and if there is some discrepancy, it sends a strong signal, a strong error signal here. It sends a strong error signal here through these climbing fibers, and what this error signal will do is that this error signal will, okay, let me explain this thing to you, that this error signal coming with the help of this olivocerebellar pathway, and it is directly synapsing with a single Purkinje fiber. It is directly synapsing with single 1 to 5 single or up to 5 Purkinje fibers. And by the way, mossy fibers, it can stimulate a lots of Purkinje fibers with the help of these parallel fibers. But here it is much more specific. As Dr. Najib says, that climbing fibers are much less promiscuous, while mossy fibers are much more promiscuous. So anyhow, these are very specific type of fibers. So as soon as there is error signal, what will happen that these Purkinje cells, they are stimulated, and they are stimulated very strongly. What I mean by this is that, a single Purkinje fiber, they have lot of synaptic connection. Their dendrites have lots of synaptic connections with the climbing fibers. So they are strongly stimulated, and as they are depolarized, they form a complex spike. What I mean by this is that there is single spike, and then there are lots of spikes like this. But if they are stimulated, if Purkinje cell, if it is stimulated by the mossy fibers, it forms a single spike, that there is a depolarization wave like this. That's it, a single spike. This is called simple spike. So this strong error signal, it started a complex spike here, and this complex spike, what it did is that it strongly inhibit the deep cerebellar nuclei, and this strong inhibition, what it will do is that it will stop the movement here. Number one, it will stop the movement here because there is the error, there is discrepancy, right? There is it is comparing the actual movement from the spinal cord. It knows that this is the actual movement. It is comparing the actual movement with the intended movement, and if there is discrepancy, if there is error, if there are significant discrepancy between actual and intended movement, the error signal is initiated. This error signal will strongly inhibit, with the help of complex spike, it will strongly inhibit the deep cerebellar nuclei, and number one, it will stop the movement immediately, because this is erroneous movement. It should not continue. It will stop the movement. Secondly, it is involved in motor learning. How it happens that, for example, if this Purkinje cell, it is repeatedly stimulated with a complex spike created with the help of this climbing fiber, what will happen that this Purkinje cells, its connection, its synaptic connection with the parallel fibers will decrease. It will decrease. So it is long-term depression. Long-term, it is called LTD. There is long-term depression of Purkinje cells. What I mean by this is that this movement, this type of movement is erroneous. So it knows, the cerebellum knows that this type of movement is erroneous. So this type of movement should not continue. So motor learning, in the sense of motor learning, these type of Purkinje cells, which were involved in this specific movement, they will sort of depress. In the long term, they will sort of depress. So that is the basic idea. Of course, it's not complete. There are certain flaws in this explanation. By the way, this explanation of this theory, this hypothesis of inferior role of inferior olivary nucleus, it is called comparator hypothesis of inferior olivary nucleus. Comparator hypothesis. Actually, there are lots of hypotheses to explain the cerebellum, but one of the important one is this that I have explained. Interestingly, as you remember that as the mossy fiber, as they were ascending upward to reach and synapse with the granule cell, they gave a collateral branch to the deep cerebellar nuclear cell. Here, the climbing fibers, they also do something similar. As they ascend, as they are ascending upward, as they are ascending superficially to reach towards the Purkinje cell, they are also giving a collateral branch, a stimulating collateral branch to the deep cerebellar nucleus. So as Dr. Najib says, then both of these fibers, as they ascend upward, they say hi to the deep cerebellar nucleus. Now unfortunately, we do not know what is the function of this collateral branch in case of climbing fiber. We are not aware of the function of this collateral branch, that their function is totally unknown. But regarding the mossy fiber, we know their function. And by the way, what is the function of those inhibitory cells that we have talked about? The basket cells, the stellate cells, and the Golgi cells, what they are doing. Okay, let me explain those cells one by one. So you see that we had those Golgi cells here, and Golgi cells, they were taking they were connecting at one end, they were connected here with these parallel fibers, right? And at the other end, they had these foot processes inhibiting at this part. Now what they do, okay, let me explain this thing to you, that the mossy fibers, they stimulated this is the granule cell. So as the mossy fiber stimulated granule cell, it here bifurcated into parallel fibers. At the same time, it stimulated the Golgi cell here. It stimulated Golgi cell, and then Golgi cell, it inhibited at this point. You see, at this point, it inhibits the connection of mossy fiber at the site of synaptic connection of mossy fiber with the granule cell. So here it released inhibitory factors at this point. So this is actually the feedback inhibition, that it prevents the excessive stimulation. It prevents the Golgi cells, they prevent the excessive stimulation of the granule cells by the mossy fibers. So mossy fibers stimulated granule cells, and granule cell in turn inhibit this connection with the help of these Golgi cells. So Golgi cells are inhibitory. Now talking about the basket cell and cells and stellate cells, they have a bit of similar function. That, for example, here is another Purkinje cell, then here is, okay, let's remove this part. So here is another Purkinje cell. So if this Purkinje cell is stimulated, the other one in its vicinity, this one and this one, they must be inhibited. Why so? To form a proper strong signal in the middle, and to prevent the noise in this area, the background noise in this area. So here what will be happening, that what is that, stellate cell will be stimulated in this region, and the stellate cell will inhibit these and this is, by the way, called lateral inhibition. And also the basket cells in this area, they will also be stimulated. So basket cell will also be stimulated, and they will cause lateral inhibition of other Purkinje cells in this region. Now that is, of course, very basic idea of these inhibitory cells. The details will be taken up in the neurophysiology lectures. And most of this information I have taken it from the Berne and Levy physiology textbook. It is also discussed partially in Guyton and some other textbooks as well, but most of the stuff there, it is better described, I think, in my opinion, it is better described in Berne and Levy physiology textbook. That is for reference. If you want to learn more, you can study that textbook. So that was about the physiology and histogenesis of cerebellum. Thank you so much for watching this video.